you for joining the final installment of the 3Rs SLG Reproducibility webinar series. This webinar is presented by the Reproducibility Working Group and is sponsored by both the 3Rs Translational and Predictive Sciences and Statistics Leadership Groups. Today's webinar is entitled A Coordinated Approach for Preclinical Research in Pharma. Our presenter is Alan Olszynski and our moderator is John Rigney, both of GSK.
My name is John Rigney and I work in the Office of Animal Welfare, Ethics, Strategy, and Risk at GSK, and I'm happy to introduce today's speaker, who's one of my colleagues in the Office of Animal Welfare, Ethics, Strategy, and Risk at GSK, Alan Olszynski. Alan received his Bachelor of Science of Biology from King's College in Pennsylvania and his Master of Science in Immunology from Hahnemann University, located in Philadelphia, Pennsylvania. During his career, Alan has been an author on over 20 publications and presented his research at local, national, and international scientific meetings. Alan is known as an in vivo biologist focusing on cardiovascular pharmacology and novel human disease targets with strong genetic associations. He has worked in academia, biotech companies, and is currently working at GSK in Collegeville, Pennsylvania, where he is an associate director in the Office of Animal Welfare, Ethics, Strategy, and Risk. In this role, he investigates and implements 3R's best practices for improving the rigor and robustness of in vivo studies. This includes work on sex as a biological variable, as well as scientifically reviewing internally and externally sourced in vivo studies, maximizing the models used and suggesting replacement of animal models where possible. Alan also volunteers as a judge and mentor at local and international student science fair competitions and is a board member for the Pennsylvania Society of Biomedical Research. With that, I turn it over to my colleague, Alan Olszynski.
Thank you, John, for that introduction. I'm just going to select the screens to share here. Presentation. And let me just check, John, you can see the presentation screen fine?
You need to flip to the presentation here in presenter mode.
Okay. Swap
there you go.
Okay. Sorry about that. Okay. So hi, my name is Alan Olszynski, and I'm at GSK, and within our group, we drive strategic approach to ensure the most translationally relevant animal models are used in our work. And this is all towards the understanding of an improvement of the translational relevance in our experiments. And what we focus on doing is to try to have a positive impact on clinical attrition rates, impact cycle times for drugs moving through the organization, have an observable decrease in animal numbers by eliminating development and refinement of low-value models, and importantly, creating 3Rs opportunities. So in this talk, I will cover 3 key aspects of developing a coordinated animal research strategy in pharma. The first topic will be model selection. And for this, I will highlight a publication from our group describing an approach to gather information on models for the purposes of having a more informed decision-making process. The second topic will cover a robust study design and best practices before the initiation of studies and what we have done internally to promote that. And finally, I'll describe a process which we call independent scientific review, which acts as a check step before study initiation and incorporates the expertise of subject matter experts across our organization. The 3Rs principles guide and underpin all of the research we do at GSK. We're committed to aligning our strategy for animal work with societal expectations of meeting regulations, performing well-designed studies, practicing a culture of care and concern for animals, and vigorously pursuing advances in science that allow us to maximize the value of these models when we use them or replace animal models where possible. And we refer to the contemporary definitions of the 3Rs for guiding all this work. And as most of you, I'm sure, already know, in this call, replacement refers to accelerating the development and the use of predictive and robust models and tools based on the latest science and technologies. Reduction refers to appropriately designed and analyzed animal experiments that are robust and reproducible and add to the knowledge database. And refinement includes new in vivo technologies which minimize the pain or distress experienced by animals, as well as a range of methods to deliver enhancements in animal care, housing, and handling. And this has led to a greater focus in GSK on leading improvements in animal care and welfare with study outcomes and reproducibility. And we encourage all of our animal care and scientific staff to bring forward more ideas about refinement opportunities through enrichment, husbandry, handling, and the environment. We have some other programs. I won't go into a lot of these, some other efforts. One we call the Model Catalog. It's a searchable spreadsheet which contains information provided directly by in vivo scientists across the organization, and it captures the purpose of the animal models, context, and use of and insight into experimental features. And it's intended to share as a knowledge database with the in vivo research community. A project that I've been working on in the past and continue is SABV, sex as a biologic variable. And it really emphasizes the importance of understanding the role of sex and that the animal has on PK/PD, disease and efficacy, and more importantly, how this translates to the clinic. And a lot of this is also supported by a need for education of the scientists on basic and more complex statistical concepts, upskilling in statistics across the research community to help understand the why of study design. So I want to make it clear that the intent of improving the animal research is not necessarily about policies and procedures, but building scientific quality across all parts of the work that we do. As we go through this presentation, you will recognize several established resources which we incorporate and refer to in the building of the animal research strategy. Our strategies are aligned with the NIH's advisory committee, to the director of the ACD working group on enhancing rigor, transparency, and translatability in animal research. And if you've seen this, you'll know that there are 5 themes in the presentation, and we try to incorporate improving study design and analytic rigor by enhancing statistical training, encouraging prospective documentation of study designs and analytical plans for our in vivo work, improving relevance and use of animal models by establishing a framework for investigators to explain the rationale for the chosen model, and setting expectations for documentation. We encourage all scientists to refer to the ARRIVE guidelines when designing a study. These lay out principles or checklists to improve the reporting of the research involving animals. We follow the ALCOA CEA principles: attributable, legible, contemporaneous, original, accurate, complete, consistent, enduring, and available. And we require all studies to have protocols written and reviewed before the start of the experiment. These would be recorded in an electronic data system and version controlled during their life cycle. And this improves research and rigor by recording hypothesis, creating transparency, and reducing the risks of bias, and incorporating statistical concepts. And finally, the prepared guidelines, which can be found in the ORCOPA website. This promotes animal alternatives and increases the reproducibility of research and testing. This is the planning, research, experimental procedures on animals, recommendations for excellence, and focuses on formulation of the study, dialogue between the scientists and the animal facility, and quality control of the components of the study. So the first topic I will talk about is model selection. And for this, I will highlight a publication from our group describing an approach to gather information on models. but for the purpose of having a more informed decision making. And this is the publication I'll mainly refer to for this first part here. So it's called Animal Model Quality Assessment, and it's a tool that we have developed to help investigators in selection of their animal models and to aid in decision makings by increasing transparency and translational quality of animal-derived evidence. So the key objectives of this part of the presentation are to familiarize you with this tool, the Animal Quality Assessment Tool, or the AMQA, and we'll look at the potential value of the tool, other parts of the GSK organization which complement the use of this tool, situations where this tool is beneficial. We'll also take a closer look at the tool, which is a questionnaire you'll see. And we'll talk about some of the limitations of the tool. And then I'll sort of end with a little assessment of some other publications that also use something similar to this. It's not just us who are working on this, it also refers to the outside community. So in the end, you should have an understanding of the tactics. Now, we're using the types of questions you can ask when developing your own assessment, and we hope you'll find this information useful as part of the research process. So we recognize that no model is perfect at recapitulating the human condition. So to optimize animal model selection for translation, and also contextualize the quality of the evidence from the animal model, it's useful to have a structured and rigorous approach to understand this. So the AMQA is a structured framework for assessing translational relevance. It helps define this relevance of an animal model for a specific question. It works at ensuring a line of sight from the patient to the animal, and the tool provides relevant questions to link the human disease to the animal model. We think it's an efficient way to identify areas of disagreement or contention with the author, the investigator. These discrepancies of opinions can be uncovered, discussed, and addressed. And it clearly identifies weaknesses or opportunities that might be refined in the model and can be thought of in a different way. So careful completion of this AMQA can highlight what the animal model might not be able to address, or may make the investigator to consider alternate models, both animal and non-animal. And it provides transparency. So there's qualified data produced with the model, which is important to all decision makers. So the AMQA will serve as a permanent record for the model and can be used for future targets or disease areas. And then it really acts as an independent evaluation. It can be used when there are concerns about animal models that might be biased by the investigator, such as we have always used this model, or this model has been used for a long time. This assessment will help sort of flesh out some of those issues. So the AMQA can be used with an educational purpose, which I think is very important, so you can instruct other people about the scientific value of the model that they're working with. So there's 5 questions in what we call the primary assessment, and then there's also a secondary assessment. I'll go through the primaries first, and then I'll show you what some of these questions look like. So the first question relates to the human disease understanding. And in there it asks, how well do we understand important mechanisms and pathways of the human condition we're intending to model? In other words, you can't model what you don't know. And in this second part of the primary questions, it's a comparative anatomy and physiology area. And these questions try to address how well is the biological context modeled. And in the third one, there's an alignment of important elements of the human disease. So we're asking here, are the appropriate mechanisms and pathways replicated for the question being asked? And the fourth one relates to translatability and pharmacology. How well is the model supported how well does the model support pharmacological translation in the past? And the last one is reproducibility of the model. How reproducible are the biological endpoints and responses in this model? Have they been determined in the past, and how confident are the investigators in moving these forward? And what we've decided to use as a scoring system for this, we had a lot of discussion as it was being developed, but we ended up on a semi-quantitative scoring system using sort of a stoplight or a red, amber, green, or a RAG assessment. And we know it's admittedly a subjective way to do this, but it provides some measure of the strength of the evidence. So the people use the tool, evaluate what is known regarding the specific questions, and then sort of reflect on the response in the appropriate boxes when they fill these in. So in general, red means a major weakness and or a difference that should be addressed. Amber, yellow color, some similarities, or there's a fair concordance between the animal and human, or where unknowns or known weaknesses should be noted. And green, we have determined that to mean a good concordance or similarities between animal and human conditions. And the scoring system was not designed to rank one animal model above another, but more to represent the strengths and weaknesses of a particular model in the context of its intended use, and to provide an opportunity to focus effort on filling any knowledge gaps that may arise as these are completed. So what's the value of doing an AMQA, this model quality assessment? The aim is that all models should have an AMQA. If you're putting the time into thinking about doing this, doing any type of model, how well is it put into context with everything else that is being developed along the process? So if you were to do this, start with your current models that you're involved in with your roles, or perhaps ones that are more frequently used and you have sufficient data with, or there's enough literature on. And then you could start one of these assessments that way, and they continue to build. And then another good place to start is with the initiation of new models. So this is, a key time to start this assessment. If you're just developing something, thinking about how to implement it, take the time to do a thorough evaluation. There's an increasing recognition and request by ethical review committees to use AMQAs to facilitate a harm-benefit analysis as they review submissions. Some of these questions, may cover some, more welfare issues or ethical issues, or uncover them too as they're being described. So, you could think of these two as adding some evidence to model selection. And then the AMQA can provide contextual support for components of a comprehensive review. So just this being just one part of a process of evaluating this in terms of a target or different phases of the research. So there are some limitations that we've recognized as we've put this together. So it's not a quality assessment of the protocol. This is more, before that time you would implement this, part of the model. The AMQA is also not necessarily an ethical review of the model or protocol. And that's the ethical part better, well suited for IACUCs and AWERBs. The AMQA does not take into consideration any other alternatives. It addresses a single model, but it can recommend other options, which could be investigated further. And the AMQA needs to be completed in a partnership with the investigator, with the target in mind, with any other subject matter experts, and it'll help ensure the relevant biological target pathways are considered. So we can take a brief look at the assessment tool right now. So the primary assessment is divided into 5 sections, and they're titled Human Condition, Model Physiology, Model Pharmacology, Model Disease, and Reproducibility, each with a broad overarching question. And then the second assessments consist of a more granular question related to the specific primary areas. So the primary questions are purposefully broad and, probe fundamental principles related to the concordance of the animal model and the human condition and the underlying disease pathobiology and previous experience with pharmacological responses. And then the secondary questions have a more granular probe to a more granular understanding of the disease in humans and the alignment with the characteristics of the animal model. Human disease is the culmination of several progressive events, beginning with initiation of that event, some etiology, an adaptive or maladaptive host response, and progression to injury or organ dysfunction, the pathology or pathogenesis. Therefore, it's important to replicate as much of these key events as possible in the initiation and progression of disease when modeling the effectiveness of a novel therapeutic. And there's also literature for in vivo on what's been termed like deliberate heterization, including different housing environments, conditions, ages, sexes, test time, and alternative test systems within in vivo studies. And this is proposed as an approach to improving the translation of animal studies to the real human conditions, because within the clinical trials, there's selection for diversity. So question 1 asks about the human condition, how well understood is it, which is being modeled, and then the secondary questions ask about characterization of the cause of the disease, how well characterizes the pathogenesis of the disease. Do we understand the role of the pharmacological target? How variable might the target patient population disease be with respect to demographics and genetic heterogeneity and other comorbidities? And is there a well characterized and translatable disease biomarker that may be applied to the animal studies? So some questions on variability, patient focused, and any key biomarkers that can be used early on. The second assessment question addresses the physiology of the model. Much of the research focuses on sometimes like the molecular resolution of what's to be understood for the roots of health and disease, but sometimes the bigger physiological picture is lost. For example, we have an example in the paper where fatty acid substrate for energy production in the mammalian heart is very well conserved across species, but the dynamics of that process is likely very different in humans with heart rates of about 70 beats per minute and rats with heart rates of about 350 beats per minute. So that doesn't mean it makes the rat an irrelevant model of cardiac physiology, but it does affect the human extrapolation of that cardiac biology. The next is the pharmacology. How well does the replicate any expected pharmacology in patients. Questions, the secondary ones point towards has the model been used previously, and has there any been predictive efficacy? How is it performed? And again, are there pharmacodynamic biomarkers for a pharmacological effect? And then understanding what we do not know about the human disease or condition of interest is also fundamental in the strategy for modeling the disease. And the complexity of human disease is exceedingly difficult to reproduce in a modeling system. So number 4 asks about the model disease assessment. How well is the development of the disease replicated? Can it be replicated in the patient? Is it the same cause in the animal model and humans? Is the model demonstrating or doing the same things to develop that disease? Is the time course similar? Sometimes animal models may try to speed along a pathology time course. So how does that potentially impact any understandings? How well does the model replicate the clinical outcomes? Are we using a chronic model to model a chronic disease or condition with the animal models? Or does the model have some acute disease initiation, which is unlike the human condition? It asks about the phenotypes, mechanisms, and intended study design. Does the animal model have the same study design that you would see in the clinical trial, and then also any translatable disease biomarkers? And the ultimate aim of any animal study, particularly in drug development, is to able a conclusion about the outcome. This relates to the reproducibility, or the ability to replicate reliably the normal or perturbed biology of interest. So although the biological variability to the human condition is useful, the variability can significantly challenge the ability to make a defensible conclusion for an animal study. So question number 5 asks about reproducibility. How reproducible are the biological endpoints, and are they of interest quantitatively and consistent in the model? So those are the 5 questions, the primary and secondary assessment. And here's an example of what a completed animal model quality assessment may look like if it were to be completed with the use of this red, amber, green scoring for the primary assessment questions. So this provides people with at least a glance of the model's strengths and weaknesses, and you can go back to the actual document that you fill out for the more detailed information on it. So this rapid visualization helps with understanding of the concordance of the human condition, and the assignment of specific colors should represent an integration of the secondary assessments. And any of these that show up as a red should not be scored anything higher than a yellow color above. So in this example, model reproducibility, it was believed by enough review and sufficient evidence that the animal model is reproducible, and then there are consistent quantitative endpoints in the model that can be measured. In this example, number 4 for the disease, there was a response to, is the cause the same in the animal and in the human? So that had to be answered as a red, because the animal model didn't have that same initiator of pathology. So that overall score receives no more than a yellow. And then you could work your way through. If there are more greens than yellows, then you could have a green color. If there's more yellows than greens, you end up with an amber color. And it's an easy way to see a weak, uncertain, or strong response for a particular model. So to provide a brief summary, the AMQA was developed to assess translational strengths of an individual model, particular for a biological target or pathway. The tool documents and considers and has considerations through a consistent framework, this question-based approach, and it'll help guide the investigator to key elements for appropriateness of the model. And once completed, the outcome becomes a context for studies conducted with the model. So it allowed decision makers to consider the translational strengths of evidence produced by the models. And similarly, the output can be used to represent the potential benefit or likelihood of a clinical success when utilizing a given model. So the values of the structured assessment lie in its ability to help in the translation. The primary aim of the AMQA is to help investigators in selection of their animal models and aid in decision making by providing this transparency. It can be a guide for investigators with limited in vivo modeling experience. If someone's relatively new to this, you can do this exercise and help you understand what the potential pluses and potential limitations are of the models. It acts as a prompt for some multidisciplinary partnership across an organization. So for the development of a study design. And like I mentioned, it's very transparent representation of the strengths and weaknesses, and provides an opportunity to mitigate any weaknesses through alternative modeling approaches or filling in some knowledge gaps, maybe even before further in vivo pharmacological studies are done. And it provides a quality context of evidence. So to use the tool also supports the harm benefit analysis, HBA. So is whether the analysis or assessment of whether the harm to the animals is outweighed by the benefits for an experiment. So I'll highlight just 3 other publications that we referred to when we were working on this. Like we're not the only ones who are developing these model assessments, and it's just proof that there's more and more interest across the industry, academia, biotech to do more of this. So one of the publications we came across was by Denier, and they describe methods to model evaluation based on how relevant the species is to the disease being studied, how complex the test system was that was being evaluated, how well the simulation of the disease is developed in the model, the predictive use of a quantile. So for this one, they use a yes or no response to determine if the drug was working or had an effect, and was there any measurable pharmacology, and the number of symptoms modeled in the test system. So what are the phenotypic changes? How well do they represent the human system? And then the next was a publication by Henderson, and they looked at research guidelines. So how should an in vivo study be run? And they found at that time 26 different relevant references. So in these publications, they came up with a list of 55 recommendations on the how-to for the running of in vivo experiments. And some of the most common criteria included powering, randomization, well-developed characterization of the model. And the last one I'd like to highlight too is a more recent publication by Ferreira, and they ask similar questions of the human condition, but they also ask for histological validation component. And then further, this tool incorporated a weighting system, which gives more or less importance to one category over the other. And you can see from their example and their publication, they were able to provide a weight and responses to each of the different categories. And then from that, they had an example of a graph, sort of like a spider webs graph, where the higher the score, the more translatable it could be going forward. So I think in the end, there's no perfect system, but it's important that this is being discussed more and more in the literature, in pharma, academia, biotech, and this will only help to lead to improvements in the decision making in the preclinical area. The next part I won't spend a lot of time on, but I think a lot of people on the call are very familiar with robust study design. And sort of the best practices and how we incorporate it before the initiation of the study. So there's been a growing awareness of research reproducibility within the preclinical area, and the publication by Baker and Nature in 2016 aimed at identifying what factors could be incorporated. And two of the top three responses included better understanding of statistics and more robust study designs. So the goal of what have developed here, robust study design, is really to improve preclinical study robustness and scientific rigor so the best decisions could be made. And the point of all this is to move from an inconsistent application of robust experimental design to a powered, randomized, blinded, in vivo studies supported by clear documentation. And in the end, this helps minimize bias and increase precision. The scope of this effort is for discovery, development, research studies. It's used internally, externally, but the way it's implemented allows for some flexibility across the organization. And sort of what has been accomplished through this is an upscaling of the understanding of statistical principles across research. This further education has included group training sessions, standards to make the ways of working more consistent, reference documentation, and FAQ, frequently asked questions sheets to help guide the process. And then just to highlight, blinding for studies, as we're sort of defining it internally. For it to be effective, the scientists must be unaware of assigned treatments. There must be a separate assessment of outcomes from people who do them. Randomization refers to the allocation of the treatments to the units, the subjects in the studies. It helps minimize confounding elements, and it can be done by simple or stratified randomization, and powering have enough precision to have scientifically meaningful differences. And we all know that powering doesn't mean having more subjects or more animals. The study design should consider studies that are potentially powered too high, which could potentially waste time, animals, or too low, which lead to potential false positives and cause us to run a subsequent study. So these are statistical principles which can be incorporated into scientific studies, and they need to be continually incorporated and updated as part of developing changes, cultural changes. And then just to reiterate, blinding may be done on primary endpoints. There may be more than one primary endpoint for studies. And if so, the other situation like this have to be addressed. And the purpose for blinding is to minimize bias. For the randomization, animals need to be randomly selected and assigned to sham vehicle or control groups. Animals can also be randomly assigned into holding rooms, cages, racks, order of dosing, order of assessment. And powering addresses the number of animals necessary to answer these questions. So this is an important component of how we do the research across the organization. The animal model data are important for the critical decisions we make and how we think about pathology of disease, pharmacological effects. And this all has to be done in a robust way and is just as important in the preclinical studies as it is in the clinical studies. So in this way, all the information is available to make decisions across the company. And the third part I'll just wrap up with here is something we've also developed internally. And it's called the Independent Scientific Review. So this really is a scientific dialogue to ensure individual studies are designed to address the predefined hypothesis. This will occur before study initiation, and it'll incorporate the expertise of subject matter experts all the way across the organization where relevant. So why is it important? As we've stated with some of the other components here, rigor of design of animal studies is critical to the delivery of scientific relevance and reproducibility and expected scientific benefit. So a well-designed or a robust study with well-defined primary readouts improve the benefit. It's a documented scientific level study. It's peer-reviewed, conducted with input from both subject matter experts across GSK, outside of the program, the business unit, and it must include biostatisticians familiar with the preclinical research and a clinical veterinarian familiar with preclinical research. It may also include animal care specialists, technicians, pathologists, clinicians, physicians, sample or subject analysis specialists such as DMPK or MRI specialists. And it works as a challenge for scientific relevance and reproducibility in a format designed to meet scientific objectives and apply the 3 R's principles. The information provided for this review must be of sufficient detail to allow scientific consideration of the hypothesis being investigated. And this study applies to internally our GLP, non-GLP animal studies conducted within GSK and at third parties, breeding colonies, genetically altered animals. So for anyone who initiates, monitors, or manages studies. So how is it incorporated? The independent scientific review is an expectation and included in our highest level policies on the use and treatment of animals. There's a single point of accountability, decision makers for the study, and accountability lies with them. There's been a written standard to describe the process. It's, like I said, meant for anyone who initiates, monitors, or manages the studies. And it's written flexible enough to cover special use cases, such as maybe blood or tissue collection. And what it doesn't cover are some of the sort of like the predefined regulatory studies, quality control studies, as long as these meet ethical review according to local requirements defined by pharmacopoeias. These independent scientific reviews also have guiding principles. The questions are formulated with a clear line of sight to the patients. The strategy to address the question considers both animal and non-animal approaches, with a bias towards the non-animal approaches whenever possible. These animal studies have to be scientifically justified, translatable, used for critical decision making, or to meet regulatory need, and demonstrate a 3 R's consideration for advancement. These animal studies also should have designs which apply high standards for reproducibility. These include methods for minimizing bias, randomization, blinding, and power, and appropriate statistical methods and robust endpoints. What else does it cover? Scientific study objectives are clearly written and achievable. Animal welfare has been considered as a component. And then there's a clear rationale for the choice of species, strain, sex, age, and from clearly reference previous work, whether that be in silico, in vitro, or in vivo. And the protocols are in a suitable electronic format, as defined by the business. So we end up classifying these studies for review by maybe three different sort of categories. There's bespoke or complex studies. This refers to a one protocol, one document describing a study. These are like standalone studies for review. There's a repeated study design. These are like a template, parts of protocols that can be duplicated. These include some PK studies with minor variations. And then the mechanism for review. So we have we have some flexibility in this too. So a standing review forum. So it's recommended particularly for complex or bespoke study designs. Again, there must be some documentation and required elements. But electronic reviews are also suitable for the reviews. These can be, maintained in some shared file structure. And existing review mechanisms may cover requirements of this standard. And although the mechanisms may be different, the output should be the same. But again, all this has to be documented evidence of a review, including the stated core values, which we previously mentioned. So sort of as an overall summary about the coordinated approach preclinical pharma that I've tried to get through today, I've only shown three small parts of what we do here, but this is a lot that's been worked on recently. So sort of summarizes, animal work remains a vital component of our drug discovery process and development, and is it's still mandated by law for certain purposes, such as non-clinical safety and vaccine batch release. The animal research strategy challenges contemporary approaches so that it can maximize the effectiveness of both animal and non-animal approaches. And we're committed to aligning our strategy for animal work with societal expectations, regulations, performing well-designed studies, practicing a culture of care and concern where possible. And I have described what I have described not only aligns with existing best practices, but also sometimes exceeds these expectations. Provided a rationale for development of the animal model quality assessment tool. The questions for the assessment are a high-level scoring system for defining predictive translatability. We've gone through what we do and for the robust study design, including decision making, for robust data, which results in improved reporting of studies tied to reproducibility. It's aligned with the research integrity and supports the judicious use of animals and resources. and also incorporates upskilling on sound statistical principles, which are incorporated into the scientific disciplines. And then the last part was the independent scientific review, which challenges individual studies for scientific relevance and reproducibility, and must meet the scientific objectives and supported by documentation and a clear line of sight to our patient. And then all of this is underpinned by the 3 R's of replacement, reduction, and refinement. And we have defined high-level targets for each of these R's, and a new 3 R strategy constructed to focus on opportunities to drive these targets. So I know we're close to time here, but I'd like to thank at the end here Gina Wilkerson, who's our new Chief of Animal Welfare Ethics Strategy within GSK, Joanne Story. She is my manager and executive director of the Animal Research Strategy and Global 3 R's. Maria, also within our Animal Research Strategy Group, scientific director. I mentioned some of her important work on the animal or the model catalogs. John Rigney within our group, who does a lot of pulling all these parts together and organizing for everyone to see through our sort of internal web pages and compiling information from different workstream projects. Mandy Berquist, who's a senior statistics director, very instrumental on the robust study design and upscaling of the statistics, and Christine Schnackenberg from the Novel Human Genetics, who has worked tirelessly also with a lot of these efforts in conjunction with our group and with the statistics part too, to drive a lot of these things forward. So I'll thank you for your attention, and I'll take any questions we have here.
Thanks, Alan. That was a good presentation. We do have a couple questions that were in the chat. If anybody else has any, you can hit the three little dots in the right-hand corner, and there's a Q&A section. You can put your question in there. But first question is from Brianna, and she asks about the AMQA. So she's curious how the AMQA tool is used when evaluating a new model altogether, where you may not be able to answer all of the questions.
Yeah, so I think that the purpose of those are to try to get as much information as you can, and the point there too was to where are those gaps? Is it a gap in the model? Is it a gap in information? So if you can highlight those gaps, does that then help you maybe do an experiment or something to try to get at those answers? Or are there is there not enough information known that maybe you consider more complex in vitro model work first, or some smaller experiments where you can maybe fill in some of those gaps?
Okay, so the next question again is from Brianna, and this is switching gears. This is in respect to the robust study design, and specifically for blinding. She mentions that she's gotten a lot of pushback on this from scientists. Basically, they're more worried about error with blinding. She wants to know, do you have any recommendations for getting more buy-in to blind studies and minimizing the dosing error?
Yeah. So without blinding, the potential of bias increases, I won't say significantly, but it could be significantly. There are examples in the literature where, blinding has not been done. Assumptions on hypotheses, conclusions have been made, and you put yourself more at a risk of having either to repeat a study or progress something that goes on to a clinical trial where now you're involving humans, and potentially a lot more money, and you end up in a spot where a lot of this, it turns out to be not only a waste of animals, but funds from organizations. So if you look at it that way, I think that the risks of not incorporating blinding are just tremendous, because everyone has some potential bias in them, whether they I like to say that accept it or not, but incorporating as much of that blinding as possible is key. And you can start out by, what is the primary endpoint? Let's blind that. So you don't have to blind an entire study. You don't have to blind all the animals and all the rooms and all the endpoints. Pick one to start with, and that should be that primary endpoint. Start there and have it well, documented as to how that blinding process is going to happen. And if people understand how that process is going to happen and how the blinding key is held, and when that blinding key is allowed to be released, I think people become more comfortable with it. I know it's a struggle in that process as you move through it, but once you get past all that, I think the benefits just outweigh anything.
Yeah, and I think that's what, just to follow up on that from perspective of what I've seen, is that just having people get more comfortable with the process and change the way they work, I think is a big thing. So sometimes it takes people out of their comfort zones of how they've always done things. So, okay, moving on. We have a couple more questions here. We have one from Terry. They want to know, what is your thoughts on the IACUC involvement in the process? And I'm assuming this was in regards to the ISR because of the timestamp.
Yeah, so I know from attending other meetings too, there's been this, external meetings, there's been discussion on that. And I guess I've heard like both camps of like yes and no. It seems to me the IACUC is in a position for a lot of the ethical welfare parts of studies, what can and cannot be done and how things are to be done. And depending on the way those IACUC protocols are written, sort of like as a general of, what happens in experiments. And I know the competing sides may say, well, let's look at these on an individual study basis too. That I think that it can be done, but that's potentially, unless the IACUC is organized differently to take on some of that workload of the number of studies, depending on what type of organization. If it's potentially a pharma company, there may be way too much to handle that way. Maybe for some smaller organizations, it can be done that way. But then you have to pull into the IACUC, the subject matter experts for a particular disease or a particular, pharma model or something. So I'm aware that there's some back and forth on this right now.
Okay, and then in the interest of time, because we are up against time here, but there was one more question. And I'm sorry if I get your name wrong. Is it Theory? They ask, one of the challenges is to change the mindset of the scientists, regulatory affairs, project team members, and to move away from usual models. How do you manage that?
Yeah, so that's from a bottom up and then top down. You can't have just your investigator, your scientists, your bench scientists, or those working in the facility say this is what we're going to do. It has to be, it could, an upswelling can start from there. It has to move up through management. It has to, sort of make its way to the top. Plans have to be put in place of, this is what we want to do. This is why we want to do it. These are the benefits of doing it that way. And then sort of the comeback back down. But everyone has to buy into it, from the people working in the labs to the managers to senior management. It has to be sponsored all the way, incorporated and sponsored all the way up and down.
Okay. Thank you everyone for attending, and Alan, thank you for presenting today. On behalf of the IQ, we will see you in the next webinar. And any you should be seeing this recording and contact information and things like that available if you have any further questions for Alan. So with that, we'll close out the webinar, and want everybody have enjoy the rest of their Thursday.
Thank you.
All right. Bye.
Thanks, everyone. Take care.